Previous class we discussed about carbohydrate, what is carbohydrate, what are the different types of carbohydrate and what are the examples. Now I will discuss about amino acid. Amino acids are the organic compound which contain both amino group and another is your carboxylic group. If an organic compound contains bond NH2 and bond COH, then such type of organic compound are considered as the amino acids. So we can write the organic compound which contain both amino group, that is your bond NH2, and carboxylic group, that is your bond COH, are called amino acid. The amino acid can be classified in different way. So next I will discuss about the classification amino acid. Basing upon the position of amino group, that is your bond NH2, with respect to carboxylic group, the amino acids are classified into different types such as number one is your alpha amino acid, number two is your beta amino acid, number three is your gamma amino acid, number four is your delta amino acid. Now I will discuss about the alpha amino acid. Take an example of alpha amino acid, NH2 bond CH2COH. This is said to be an alpha amino acid because the NH2 group is attached to the alpha carbon with respect to this functional group. The name of this alpha amino acid is your glycine. Similarly, you can take another example, NH2 bond CH, bond CH3, bond COH. Look at that, this is your carboxylic group and this carbon is considered as alpha carbon with respect to this group, and this is your said to be beta carbon with respect to this COH group. Now the NH2 is attached to the alpha carbon, so this type of amino acid belongs to the alpha amino acid, and the name of this alpha amino acid is your alpha alanine. From the example, we can conclude that in alpha amino acid, the NH2 group is attached to the alpha carbon with respect to the carboxylic group. So we can write, if the bond NH2 group is attached to the alpha carbon atom with respect to bond COH group in an amino acid, then such type of amino acid are called alpha amino acid. Next I will discuss about the beta amino acid. Take an example of beta amino acid, NH2 bond CH2 bond COH. This is your said to be carboxylic group and this is your said to be alpha carbon with respect to this functional group. This is said to be beta carbon, and NH2 is attached to the beta carbon with respect to the COH group, so it belongs to your beta amino acid, and the name of this compound is your beta alanine. From this example, we concluded that if the bond NH2 group is attached to the beta carbon atom with respect to the bond COH group in an amino acid, then such type of amino acid are called beta amino acid. Next, I will discuss about the gamma amino acid. Take an example of gamma amino acid: NH2, CH2, 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 COH. This is your said to be alpha carbon with respect to the carboxylic group. This is your beta, this is your gamma. Now the NH2 is attached to the gamma carbon atom, so it belongs to your gamma amino acid, and the name of this compound is your gamma amino butyric acid. From this example, it is clear that if the bond NH2 group is attached to the gamma carbon atom with respect to the bond COH group in an amino acid, then such type of amino acid are called gamma amino acid. Next, I will discuss about the delta amino acid. Take an example of delta amino acid. Take an example of delta amino acid: COH, CH2, CH2, C double bond O, bond CH2, NH2. Look at that, with respect to this bond COH group, this is your alpha, this is your beta, gamma, this is your delta, and the NH2 group is attached to the delta carbon, so it belongs to your delta amino acid. The name of this compound is your delta amino libulinic acid. From this example, it is clear that if the bond NH2 group is attached to the delta carbon atom with respect to the bond COH group in an amino acid, then such type of amino acid are called delta amino acid. Amino acid can be classified depending upon the relative number of bond NH2 and COH group in the molecule. So we can write basing upon the relative number of bond NH2 and bond COH group present in the molecule, the amino acid is classified into three types. Such are: number one is your neutral amino acid. Number two is your acidic amino acid. Number three is your basic amino acid. At first, I will discuss about the neutral amino acid. In neutral amino acid, the number of bond NH2 group is equal to the number of bond COH group. As a result, the basic strength given by the bond NH2 is equal to the acidic strength given by the bond COH group. That's why the molecule is considered as a neutral, and this type of amino acid belongs to your neutral amino acid. So we can write in this type of amino acid, the number of bond NH2 group and number of bond COH group are equal. The examples of neutral amino acid are glycine, alanine, valanine, leucine, isoleucine, serine, threonine, cysteine, methionine, phenyl, aniline, triosine, tryptophan, proline, asparagine, and glutamine. These are the example of neutral amino acid. Next I will discuss about the acidic amino acid. In acidic amino acid, the number of bonds UH group is more than the number of NH2 group. So we can write in acidic amino acid, the number of bond NH2 group is less than the number of bonds UH group. Example of acidic amino acid is glutamic acid, aspartic acid. These are the example of acidic amino acid. Next I will discuss about the basic amino acid. In basic amino acid, the number of bond NH2 group is more than the number of COH group. So we can write in basic amino acid, the number of bond NH2 group is more than number of bonds COH group. The examples of basic amino acid are histidine, lysine, arginine. These are the examples of basic amino acid. The amino acid also classified basing upon the nutritional requirement. So we can write basing upon the nutritional requirement amino acids are classified into two types. Such are, number one is your essential amino acid. This is also called indispensable amino acid. The second is your non-essential amino acid. It is also called dispensable amino acid. At first I will discuss about the essential amino acid. The essential amino acid must be supplied to our body through diet, but the non-essential amino acid already produced in our body, so it is not necessary to supply the amino acid to our diet. So we can write these are the amino acid which is supplied to the body through diet for the maintenance and growth of our body, because because these are not synthesized in our body. The examples of essential amino acid are valine, leucine, isoleucine, phenyl, alanine, arginine, histidine. These are the examples of essential amino acid. This type of amino acid must be supplied to the body through diet. Next, I will discuss about the non-essential amino acid. This type of amino acid is synthesized in our body, so it is not necessary to supply this type of amino acid to our body through diet. So we can write these amino acids are synthesized in our body and need not to supply to our body through diet. The examples of non-essential amino acid are glycine, alanine, triosine, serine, cysteine, aspartic acid, asparagine, glutamic acid, glutamine, and proline. These are the non-essential amino acid. This is all about your classification of amino acid. Next, I will discuss about the function of essential amino acid. Function of essential amino acid. First is your phenylalanine. Phenylalanine is used to boosting the memory power and maintain a healthy nervous system. So we can write boosting memory power and maintain a healthy nervous system. Next is your valine. It is responsible for the growth of muscles. Next is your isoleucine. Plain vital role in the synthesis of hemoglobin, and it is the major component of RBC. Next is your leucine. It is responsible for the synthesis of growth hormone. Next is your lysine. This is responsible for synthesis of enzymes and other hormones. Next is your histidine. It is responsible for the production and synthesis of RBC and WBC. Production and synthesis of both RBC and WBC. Next is your methionine. It is responsible for a good and healthy skin. So we can write maintaining a example, in maintaining our appetites. Next is threonine. It is used for the functioning of immunity system, that is your. These are the essential amino acid and its a function. Next I will discuss about the function of non-essential amino acid. First is your proline. Regeneration of new skin. Proline responsible for the regeneration of new skin. Next is your aspartic acid. It is responsible for the synthesis of other amino acid. Synthesis of other amino acid. Next is your asparagine. Asparagine is used for the formation of purines and pyrimidines for DNA synthesis. Next is your serine. It is responsible for the growth of muscles. Next is your tyrosine. It is used for the synthesis of T3 and T4 thyroid hormones. Next is your arginine. It is responsible of biosynthesis of protein. Next is your alanine and glutamate. It is responsible for the removal of toxic substance from our body. Next is your cysteine. It function as an antioxidant and protect our body from radiation and pollution. Also, it inhibit the growth of hairs, nails, etc. It also provide resistance to the body, to our body. These are the non-essential amino acid and its function. This is all about your amino acid. Next I will discuss about the polypeptide. The polypeptides are formed by the condensation of amino acid molecules. So we can write the polypeptides are formed by the condensation of amino acid molecule. The peptides may be dipeptide, tripeptide, oligopeptide and polypeptide. So we can write the peptide may be dipeptide, tripeptide, oligopeptide and polypeptide. Take an example. of dipeptide tripeptide. Suppose I want to form a dipeptide by the help of two amino acids. One is your glycine, another is your also glycine. The structure of glycine is written like this. This is the structure of glycine. The structure of another glycine molecule is written like this. When they undergoes condensation, there is a loss of an water molecule like this, and the polypeptide which is formed is represent like this. This is said to be an dipeptide. This is said to be dipeptide. Here this amide linkage is called peptide linkage, and this is said to be dipeptide. The name of this compound is glycine. This name is given by following these two names. Take another example. Suppose I will take one glycine molecule. Another is your alpha alanine. The structure of alpha alanine is like this. And take another glycine molecule. Here 3 amino acid is taken. This is also glycine. This is also glycine. Here a tripeptide is formed. The structure is written like this. This is the structure of a tripeptide. The name of this tripeptide is your glycine. Look at that, your ion is removed, YL is attached, your ion is removed, YL is attached, and finally we write glycine. That means glycine, alanine, glycine. This is a tripeptide. Here we can write a point. The amide linkage, that is your bond NH, bond C double bond O, in peptide is called peptide linkage. Remember an important point: the number of peptide linkage is less than the number of amino acid used for the formation of this polypeptide. So we can write the number of peptide linkage in a polypeptide are less than the number of amino acid used for this polypeptide. Look at that, here two peptide linkages are present, but the number of amino acid molecule is your 3. So we can conclude that the number of peptide linkages in a polypeptide are less than the number of amino acid used for this polypeptide formation. In a polypeptide there are two terminal, one is your N terminal, another is your C terminal. This is your said to be N terminal, this is your said to be C terminal. This is your said to be N terminal, this is your said to be C terminal. So we can write in a polypeptide there is a free bond NH2 group at one end called N-terminal, while a free bond COH group at other end called C-terminal. This is all about polypeptide. Now I will discuss about protein. Protein are the nitrogenous complex compound which are found in all living matter. So we can write these are the complex nitrogenous polymeric substance present in all forms of the living matter. Protein are formed by the help of alpha amino acid. When the alpha amino acid undergoes condensation, then protein is formed. So we can write the protein is formed by the condensation of alpha amino acid. Protein can be classified in different ways. Generally we think about the classification of protein on the basis of their composition, and another is your classification of protein on the basis of their function. So we can write protein can be classified on the basis of number one is your composition, number two is your function. At first I will discuss about the classification of protein on the basis of their composition. So we can write on the basis of the composition, proteins are classified into three types, such as number one is your simple protein, number two is your conjugated protein, number three is your derived protein. At first I will discuss about the simple protein. The simple protein when undergoes hydrolysis gives alpha amino acids. So we can write simple protein on hydrolysis produces only alpha amino acid. The simple protein is made by the chain of amino acid which are joined by peptide bond based upon the three-dimensional shape. The simple protein is classified into two types. One is your fibrous protein, another is your globular protein. So we can write based upon the three-dimensional shape, the simple protein is classified into two types. such as number 1 is your fibrous protein, number 2 is your globular protein. At first I will discuss about the fibrous protein. The fibrous proteins are thread-like molecules which tend to lie side by side. So we can write the fibrous proteins are thread-like molecule which tend to lie side by side to form fiber. In fibrous protein, the molecules are held together by hydrogen bond. So we can write in fibrous protein, the molecules are held together by hydrogen bond. The fibrous proteins are insoluble in water and they resist to digestion. So we can write these are insoluble in water and resistant to digestion. The examples of fibrous protein are collagens, keratins, elastin, myosin, osin, prolamine. These are the examples of fibrous protein. Next I will discuss about the globular protein. In globular protein, the molecules are present in folded-like structure and they tend to form a spherical shape. So we can write the molecule in globular protein are folded into a compact unit and often approach to a spheroidal shape. In globular proteins, there is a small intermolecular force of attraction between the molecule due to small area of contact between the molecules. So we can write the intermolecular force in them are very weak due to small area of contact between the molecules. The globular proteins can dissolve in the water and they can digestible. So we can write these proteins are soluble in water and digestible. These proteins act as an enzyme and also act as an antioxidant. So we can write these protein function as an enzyme and act as an antioxidant. Examples of globular protein are insulin, albumin, globulin, examples of globular protein. Now I will discuss about the conjugative protein. Conjugative protein. When the conjugative protein undergoes hydrolysis, then it gives a alpha amino acid with a non-protein part. The non-protein part is called prosthetic group. So we can write conjugative protein yield a alpha amino acid and a non-protein material called prosthetic group or conjugating group, or it is also called cofactor. The main function of the conjugative group is to control the function of the protein. So we can write the main function of conjugating group is to control the biological function of the protein. Now I will take some examples of conjugative protein and a prosthetic group. Name of protein and another is your prosthetic group. Nucleoprotein is a conjugative protein. Here the prosthetic group is nucleic acid. Next is your glycoprotein. Here the prosthetic group is carbohydrate. Generally think about the sugar carbohydrate. Next is your lipoprotein. Here the prosthetic group is your lipid like lectin. Next is your phosphoprotein. Here the prosthetic group is your phosphoric acid residues. Next is your chromoprotein. Here the prosthetic group is your some coloring matter. Next is your metalloprotein. Here the prosthetic group is your some metal. These are the examples of conjugative protein. Next I will discuss about the derived protein. These type of protein are generally formed by the partial hydrolysis of simple protein and conjugative protein. So we can write these are the degradation product obtained by the partial hydrolysis of simple protein and conjugative protein. Examples of derived proteins are proteases, peptones, polypeptides. Proteins are also classified basing upon the function. So we can write basing upon the function of protein. The proteins are classified into different types. At first I will discuss about the structural protein. These are the fibrous protein. So we can write These are the fibrous protein such as collagens of cartilage and bones, keratins of hairs, nails and skins. Next is your contractile protein. These are found in muscles. Example of contractile protein is your myosin. Next is your enzymes. These are used as a biocatalyst. So we can write proteins of this group serves as a biocatalyst. Examples of enzyme which act as a protein is your trypsin, pepsin. Next is your hormonal protein. Protein of this group act as a messenger, so we can write protein of this group acts as a messenger. Example is your insulin, growth hormone. Next is your transport protein. Transport protein is a protein that serves the function of moving other material within an organism. Examples are hemoglobin, serum, albumin. These are the examples of transport protein. Next is your storage protein. This type of protein serves as biological reserve of metal ions and amino acid used by organism. Example is your OVA, albumin, glutatine. These are the example of storage protein. Next is your genetic protein. These proteins generally related to genes. Example is your nucleoprotein. Next is your defense protein. The antibodies are the defense protein. Example is your immunoglobulin. Next is your receptor protein. These proteins receive the chemical signal, and the examples of these protein are guanine, serine, threonine, kinases, etc. This is the classification of protein depending upon their function. Next I will discuss about the structure of protein. The structure of protein is quite complex. Therefore, the structure of protein is discussed in 4 levels. Therefore, the structure of protein discussed in four different levels, such as number 1 is your primary structure, second is your secondary structure, third is your tertiary structure, and fourth is your quaternary structure. Now I will discuss about the primary structure of protein. The primary structure of protein is drawn like this. This is the primary structure of protein. This is said to be peptide linkage. This is one amino acid unit. This is the another amino acid unit. The primary structure refers to the sequence in which various amino acids are linked together by the peptide linkage. This is your said to be peptide linkage. The primary structure of protein is determined by the successive hydrolysis within acid, alkali or enzymes. The successive hydrolysis of protein yield different product having decreasing molecular mass as shown below. Suppose I will take a protein and it is subjected to successive hydrolysis, then what are the different products obtained? Look at that. When protein undergoes first hydrolysis, then it gives proteoses, which again hydrolysis gives peptones, again hydrolysis gives polypeptides, which again hydrolysis gives simple peptides, which again hydrolysis gives alpha amino acids. Protein, when hydrolysis gives proteoses, which on hydrolysis gives peptones, which undergoes hydrolysis gives polypeptides. Next, when polypeptide undergoes hydrolysis gives simple peptides. Now simple peptide on hydrolysis gives alpha amino acid. In primary structure, the peptide bond acts as a backbone and the side chain, and this side chain project outside the peptide backbone. So we can write in primary structure the peptide bond form the backbone and the side chain project outside the peptide backbone. Now I will explain about the secondary structure of protein. There are two secondary structure of protein are possible depending upon the size of R group. Such as number 1 is your alpha helix, number 2 is your beta plated seed structure. Now I will discuss about the alpha helix structure. Remember an important point. In alpha helix structure, the R group having larger size, but in beta plated seed structure, the R group having less size. So we can write when the R group size is more than intermolecular hydrogen bond is formed between C bond O group of one amino acid and bond N has group of another amino acid present in the chain. As a result, the polypeptide coils up into a spiral structure called right-handed alpha helix. Now I will draw the alpha helix structure like this. Look at that. How can I arrange the C double bond O and NH group in this spiral structure? This is your C double bond O. Here nitrogen atom is written and hydrogen bond is shown here. And here C double bond O is written. Now here nitrogen atom is written. Here C double bond O is written. Suppose here nitrogen is written and hydrogen is written like this. Suppose I will draw the carbon here and the double bond oxygen is written here. There is a formation of hydrogen bond. Suppose here nitrogen atom is present, here hydrogen bond is present. Suppose here C double bond O is present, here nitrogen atom is present. Suppose here C double bond O is present, there is a formation of hydrogen bond this. Suppose here C double bond O is present, there is a formation of hydrogen bond this, oxygen and hydrogen. You can also form hydrogen bond like this. Here hydrogen bond is also formed like this. This is the alpha helix structure. The alpha helix structure seen in fibrous protein like alpha keratin, in hair, nail and myosin in Next I will discuss about the beta plated sheet structure. When the size of R group is small, then the polypeptide chains lie side by side in a zigzag manner within alternate R group on the same side situated at a fixed distance. As a result of which intermolecular hydrogen bond occur between two neighboring chain. This results in the formation of a flat sheet structure. Look at that. Suppose I will draw a polypeptide chain like this. This is one polypeptide chain. Suppose I will draw another polypeptide chain like this. Here is a hydrogen bond formed between this oxygen and hydrogen. Here also hydrogen bond will be formed. Here a hydrogen bond formed like this. Here also hydrogen bond formed like this. This is one polypeptide chain and they are arranged in zigzag manner. This is another polypeptide chain, they are also arranged in zigzag manner. And there is a hydrogen bond formed between the two polypeptide chain. Look at that, here R is present and here also R is also present, here also R is present. These are present in the alternate manner at a fixed distance. As a result of which intermolecular hydrogen bond occur between the two neighboring chain. This results in the formation of a flat sheet structure. This is all about your beta plated sheet structure. Now I will discuss about the tertiary structure. An alpha helix may be considered a piece of rope. We know that the rope can be easily bended, folded, twisting. So we can write an alpha helix may be considered as a piece of rope which easily bended, folded, twisted. Like this, the tertiary structure of protein also easily bended, folded, twisted. So we can write the tertiary structure of a protein refers to the final 3D, that is a three-dimensional shape that results from the twisting, bending and folding of the protein helix with a main forces operating are hydrogen bond, disulfide linkage and van der Waals forces of attraction. That means in tertiary structure we generally think about the hydrogen bond, disulfide linkage, van der Waals forces. Basically, if you think about the tertiary structure of protein, generally we think about the twisting, bending and folding, and also think about the three-dimensional cell. Tertiary structure of proteins results with a fibrous and globular cell. That means it generally deals with the fibrous and globular shape of the protein. Next I will discuss about the quaternary structure of protein. Complex proteins are formed by the combination of two or more polypeptide chain. That means the quaternary structure deals with two or more polypeptide chain, and these polypeptide chain are already having primary, secondary and tertiary structure. So we can write each chain is a complete protein with a characteristics primary, secondary and tertiary structure. The quaternary structure refers about the polypeptide chain and the linkage of each polypeptide chain. So we can write the quaternary structure refers to the way in which these polypeptide chains of a complex protein are associated with each other. This is all about the structure of protein. Next I will discuss about the biological role of protein. First point is your protein act as a catalyst. Protein act as a catalyst. Second point is your the fibrous protein act as a component of tissues. Next point is your protein hormones regulate the growth of plants and animals besides controlling many other psychological functions. Interferon is a cellular protein which is naturally produced by the body in response to virus infection. Blood plasma is a solution of protein in water which is used for the treatment of shock produced by serious injuries and operation. These are the biological role of protein. Except the biological role of protein, there are another uses of also protein. It is the essential part of our food, generally found in egg, milk, fish, etc. Casein is used for the manufacture of artificial wood and silk. Amino acids present in the protein are used for medicinal uses. Gelatin is used in dessert, salad, candies, bakery goods, etc. Leather is obtained by tanning of animals' hides. Hemoglobin present in the blood responsible for carrying oxygen and carbon dioxide. Hormones control various body process. Enzymes are the protein produced by the living system and catalyze specific biological reaction. These are the uses of protein. An important question always asked in the exam: what is the denaturation of protein and what is the cause of denaturation of protein? So I will discuss about the denaturation of protein. The process after which the protein loses their physiological activity and other property, that is called denaturation of protein. So we can write the process after which protein loses their physiological activity and certain other properties is called denaturation of proteins. denaturation of protein is caused by, number one is your action of bacteria, action of heat, by shaking with the alcohol, by treating with the acid. Due to this, denaturation of protein takes place. We know a daily life example. When an egg is heated, then the coagulation of white part of the egg takes place. So we can write examples of denaturation of protein is, first is your coagulation of the white of egg on heating, coagulation of milk in the presence of an acid to form the cheese. So the second example is your coagulation of milk in the presence of an acid. Basically we use lemon juice to form cheese. Now I will explain about the cause of denaturation of protein. What is the cause of denaturation of protein? When a protein is subjected to heating or treating with the alcohol or acid, or when the protein is attacked by a bacteria, then denaturation takes place. When a protein is subjected to heating, then what will happen? There is a disruption of the secondary and tertiary structure of the protein due to the breakdown of the hydrogen bond. As a result, the protein molecules are uncoiled. That is said to be a denaturation of protein. So we can write denaturation of protein is due to the disruption of the secondary and tertiary structure due to breaking of hydrogen bond and salt bridges in protein, causing uncoiling of the protein molecules from an ordered and specific conformation into a random conformation, resulting into its a coagulation from solution. This is the cause of denaturation of protein. Next I will discuss about the renaturation of protein. Renaturation of protein is the reverse of denaturation of protein. So we can write the reversible process by which the original form of the denaturated protein can be regained is known as renaturation of protein. Remember some important point. Protein denaturation may or may not be reversible. Next point is coagulation of white of an egg on heating is an irreversible process, but in case of reversible denaturation, the protein can be coagulated from the colloidal solution by saturating the solution with a soluble salt like ammonium sulfate, or by addition of water or alcohol, thus rejoining the original form. This is all about your protein.